Hey, my name's Rachel Davis, and I'm going to be presenting a study done involving social brain activity. This lab is called Viewing Ambiguous Social Interactions Increases Functional Connectivity Between Frontal and Temporal Nodes of the Social Brain. First, I'm going to start off with some background information necessary to understanding the methods and results of this lab. Social behavior is defined as a visual perception that may affect our actions or emotions. This is the main idea that we will be centering around. We will be studying primates for this lab that are accustomed to social environments. Based on past research, we know that there's a frequency synchrony in regions during social decisions. This involves overlapping activity in regions related to understanding, processing, and acting on social cues and interactions. In this lab, there are 5 important ROIs, regions of interest, that we will be discussing. This includes the temporal lobe, which is involved with processing emotions and mostly perceiving our visual surroundings, the SDS, or the superior temporal sulcus, involved in social attention and eye gaze, the left frontal lobe, the premotor cortex, involved in selection of appropriate motor plans, the cingulate gyrus/cortex that helps regulate emotions and pain. It has also been known to help with predicting negative consequences, and the caudate nucleus that involves planning, movement, learning, memory, reward, motivation, emotions, and sometimes romantic interaction. We will also be using a technology called bold fMRI data, which helps us understand brain activity through identifying increased blood flow to a certain region. The main focus of this lab is to observe the interconnectedness of brain regions in monkeys associated with social behaviors to help identify a main network of how and when they work together. The 3 questions proposed include: how do the different regions that perceive social interactions and mental states communicate? How do these regions react when viewing single actors versus multiple actors in a social interaction? And how do these regions react when viewing different behaviors/interactions? The 3 behaviors include ambiguous, known as unclear or confusing interactions, aggressive, known as violent fighting or chasing, or affiliative, known as grooming or mounting. In the figure on the right, I'd like to point out that the clips are separated by blank areas, or the off regions, and areas of white, which are known as visual landscapes not depicting social behavior. This shows how the video was set up during the experiment, and we will be discussing it in more detail later on. We hypothesized that this widespread network synchronization occurs when cingulate and temporal areas coordinate their activity when more difficult social inferences are being made. For the methods and materials for this lab, we collected fMRI data from 3 male macaque monkeys, which are all 7 to 8 years old, weighing 10 to 13 kilograms. They all lived in large communal rooms. However, M1, monkey number 1, and M2, monkey number 2, were pair housed, while M3, monkey number 3, was singly housed. We recorded from 16 anatomical ROIs, centered in the 5 different areas discussed in the introduction. In the figure below, we understand how we picked some of those regions through GLM analysis, which is like an extraction of a signal from regions that are continuously responsive, and voxels, which are 3D images of brain tissue. The monkeys each watched a video clip 880 seconds long, with 5 to 20-second video clips of the 3 types of behavior: affiliative, aggressive, and ambiguous. It was integrated with 20-second blank clips. There are periods of no social interactions where the video depicted visual landscapes, and periods where the video was turned off. It's important to note that the actors in the video were macaque radiata monkeys, not in the same species as the monkeys being tested. Although the monkeys were freely watching the videos, they were rewarded for maintaining their gaze. We received some very convincing results supporting our hypothesis, which includes: aggressive behavior showed limited increase in temporotemporal, which, if you remember from the intro, is involved with perceiving visual surroundings and processing emotions. It also showed a weak increase in the cingulate temporal connectivity, which involves predicting possible outcomes. Affiliative behavior was mentioned to not cause significant increase in functional connectivity in any of the ROIs discussed. However, ambiguous scenes elicit increased functional connectivity in the frontotemporal connections, which involves selection of appropriate motor plans. Specifically, the activity was in the cingulate temporal and the temporotemporal, which was higher in the first several seconds. On the figure to the right, we see these results depicted, with ambiguous behavior being demonstrated by the purple line, aggressive behavior being demonstrated by the red line, and affiliative behavior being demonstrated by the blue line. As you can see, there's a clear increase in activity, mostly prominent during ambiguous behavior. When the video played, we also received data varying based on how many actors were in the video. During blank scenes, it showed temporotemporal functional connections dominated, and the frontal cortex was less pronounced. During scenes with no actors, aka visual landscapes, the temporotemporal functional connectivity was recorded. During scenes with only single actors, bilateral activation was recorded in the temporal cortex. No activation evident outside the cortex, although. During scenes with multiple actors, strong activation in the bilateral activation in the cingulate gyrus. Here are some key takeaways from these results. This suggests that communication between the cingulate and the temporal cortices enhances/increases when encountered with more difficult/confusing social situations, like ambiguous situations. This also suggests the idea that frontal temporal connectivity relies on social context. In theory, this lab could contribute to future understanding of social behaviors and possibly help scientists and therapists accommodate for certain disorders, including autism spectrum disorder, which involves a deficit in communication, social interaction, and behavioral skills; schizophrenia, which involves the prefrontal and medial temporal regions discussed in the lab, which may provide more information into the negative symptoms like delusions, hallucinations, or disorganized thinking; and social anxiety disorder, which is a fear of humiliation, interacting, and communication with others. I would also like to further discuss the figure to the right, which demonstrates centrality. A higher centrality essentially means there's a higher level of connections to the region circled. This figure demonstrates how interconnected our brain really is and how hard it is to fully understand disorders that may involve structures in these pathways. In conclusion, although the temporal lobe was activated during most of the experiment, the frontal lobe areas were only activated with more than one actor and fluctuated based on the type of behaviors viewed. Due to learning the highest activation of the frontal lobe in the cingulate cortex was during ambiguous behavior, we can take away that it might be in our nature to infer a possible outcome from behaviors and interactions. This gives the cingulate cortex more meaning relating to its range of information for making social decisions. Overall, social cognition is becoming clearer every day with more research. Here I offer some critiques of the lab that I think could be taken into consideration for future labs. I think in this lab they looked at lots of variables, including how many actors were present, which regions to look at, and only 3 behaviors. I would consider focusing on only one variable, specifically how the monkeys react to just the 3 behaviors, and possibly using more behaviors to look at. I also suggest that they have a couple monkeys watching longer clips on the behaviors to fully understand more context of the situations. The technique of the video in this lab was too separated and included too much blank area compared to the heart of the video, which is supposed to be the behaviors. I also suggest that they show videos of the same species, considering some species react differently to certain actions than others. Lastly, I suggest they use more advanced studying technology other than fMRI with higher temporal resolution to capture the brain activity data millisecond by millisecond.